Hi, I am Suraj, final year life science student at Atria University. I am working on some projects. So one of my important project is we are annotating Ralstonia solanacearum bacteria, which is a very well-known pathogen known for its adaptability and highly virulent genes. So this is also important because this has some economical factors because it affects the economical crops which are important and which could be expensive. So in this study, we are actually annotating genes. We are analyzing our genome. So we got two isolates of Ralstonia from Odisha from a university. So we got two isolates from two different species of plants. So we have started with the raw data. So we have checked the quality of the data. Then it could be the like there are softwares, many softwares. We did assembly. We have used spades, and for spades because it needs a high computing power. So I have used Google Cloud platform to utilize those softwares. So in this way, I've used many quality checks after each step. Like after assembly, I've used Quast and Busco, and after annotation, I've used Busco part and check them. There are various versions which are available. So because it includes many softwares, so I utilized Docker to package them together, and I use Nextflow also to make a full-fledged pipeline with all the softwares at once. For this project, I also supported by one of the Swiss laboratories from University of Zurich, and I will be work there on an UTR infection. So I am happy to work there, and I am also excited to work there on a human pathogen. I want to deep dive into the field of bioinformatics, and I have chosen it as my career. Thank you.